Hello, everyone, and welcome to today's panel discussion. My name is Karen Hunnity, and I'm the Director of Corporate Communications and Investor Relations at Athersys. And I'd like to thank you for joining us today to learn more about the TREASURE data. The TREASURE study was conducted by HEALIOS, our partner in Japan, and it was a randomized, double-blinded, placebo-controlled study evaluating MultiStem administration for the treatment of ischemic stroke. We have put together an expert panel of key opinion leaders in the field of neurology to discuss their own perspectives on the TREASURE data. And I want to thank each one of them for taking the time out of their busy clinical schedules to be here with us today. Dr. Robert Willie Mays, our Vice President of Regenerative Medicine and Head of Neuroscience Programs here at Athersys, will be moderating the event today. He's going to be taking questions from all of you on this webinar and directing those questions to the panelists. We will be answering all the questions verbally. So if you have a question at any time during the webinar, please type it into the Q&A window and you'll find the Q&A option at the bottom of your Zoom screen. This presentation is also being recorded and the webcast will be available on our website at www.athersys.com shortly after the event. And if for any reason we did not get to answer your question, you may also send it to ir@athersys.com after the event concludes. The views and opinions expressed by our panelists today are their own. Dr. Mays and our panelists may be making forward-looking statements, and the safe harbor rules govern our remarks we make today. These forward-looking statements relate to, among other things, statements regarding anticipated results of clinical trials, including, and the potential benefits of, our MultiStem product candidate. Forward-looking statements are subject to risks and uncertainties that could cause actual results to differ materially, including, among others, the possibility of unfavorable results from ongoing and additional clinical trials involving MultiStem, and the risk the positive results in a clinical trial may not be replicated in subsequent or confirmatory trials. The company assumes no obligation to update forward-looking statements to reflect actual results, subsequent events or circumstances, or other changes affecting forward-looking information except to the extent required by applicable securities laws. Now with that, it is my pleasure to turn the meeting over to Dr. Willie Mays, the moderator for our event today.
So thank you, Karen, and to my esteemed key opinion leaders. You guys can start to turn your videos on now. Thanks, everybody. Really appreciate everyone joining us on the call today. Time to participate in the webinar and to hear from and interact with some of the world's leading stroke neurologists, as well as one of the preeminent clinical trial statistical researchers in the United States. Recently, our partner, HEALIOS, announced top-line data results from their phase 2/3 randomized double-blind placebo-controlled clinical trial called TREASURE, which, as Karen said, evaluated the efficacy and safety of the MultiStem cell product compared to placebo in 206 ischemic stroke patients enrolled at 48 clinical sites throughout Japan. And the morning the trial results were made public, and we had a lot of press releases, both us and HEALIOS, I received enthusiastic emails or congratulatory messages from stroke neurologists, investigators from our Masters 2 clinical sites who had reviewed the press release, academic researchers we've worked with, and the panelists we've engaged today. So my hope for this roundtable discussion is that the experts we've assembled can answer your questions directly, share their perspectives, and convey to the Athersys community the same sense of excitement regarding the data from TREASURE and the potential for MultiStem to change the lives of stroke patients and their families. So just, I'll shut up here as quickly as possible. Just a few ground rules for the roundtable. As Karen said, this is completely unscripted. The panelists' comments and opinions are their own, and we hope to provide to all of you the human side of treating stroke from the perspective of these leading neurologists who see patients and their families every day. and better understand what long-term recovery and independent living could mean for those people that are affected by stroke. I will be reading questions and asking for responses from the panel. Otherwise, I'll try to be as unobtrusive as possible. And the panelists, if I don't ask you a question directly, feel free to add on to the comments and perspectives others have already shared. If you're joining live and you don't get a chance to get your question asked, you may send it to ir@athersys.com after the webinar, and we will make sure that it gets answered. And so with that, panelists, I'd like you to begin with some brief introductions. I'd like you to tell us your name, your affiliation, and any specific stroke interests or research focus you have. And I did this alphabetically. So Tom, I'm going to start with you.
Great, thanks, Willie. I'm Tom Carmichael. I'm professor and chair of neurology at UCLA. I see stroke patients clinically. I do neuro rehab and stroke recovery clinically. And I have a basic research lab that looks at cellular and molecular mechanisms of brain repair after stroke.
Thank you very much, sir. Dr. Chu.
Hello, Willie. Hello, everybody. I'm David Chu. I head the stroke research program at Houston Methodist Hospital, and I'm professor of neurology at Weill Cornell Medical College. And I'm a proud site principal investigator at Houston Methodist Hospital for the Masters 2 trial.
Great. Thank you very much, David. And we'll go to the other David, Dr. Hess.
Thanks, Willie. So I'm David Hess down here in hot Georgia, 100 degrees today, in Augusta, Georgia, Dean of the Medical College of Georgia and a stroke neurologist. I've been working with Athersys, since probably the mid-2000s, and worked in some of the preclinical evaluation. And then the Masters 1 trial was one of the primary authors. So I continue to be active in stroke research, mostly acute stroke, looking for stroke agents that are effective in people. We have a lot of them that work in rodents, but we don't have a lot of them that work in people yet.
Great. Thank you very much, David. And to prove to all the attendees today that these really are stroke neurologists, Dr. Savitz would be next, but because he is currently working to care for stroke patients, he'll pop in later when he gets a chance, but he's in the clinic right now. So we'll go to Dr. Wexler.
Thanks, Willie. So I'm Larry Wexler. I'm a professor of neurology and chair of neurology at Pennsylvania Hospital at the University of Pennsylvania, where I've been for the past couple of years. Before that, I was at UPMC for 35 years, and for the last 11 of those years, chairman of the neurology department at UPMC. Before that, the director of the Stroke Institute, which I founded way back in 1995 to help find new and better therapies for stroke. I've been involved in cell therapy for stroke since the late 90s, involved in many of the clinical trials that have been going on for the past couple of decades, including Masters. I was right after David Hess on the Masters paper, so we were all very intimately involved with that study, and look forward to this discussion about the TREASURE trial.
Great. Thank you very much, Larry. And then we're going to close with our esteemed statistician from Harvard. LJ, please introduce yourself.
Yes, thank you, Dr. Mays. It is a really great pleasure to join this distinguished panel. I am a professor of biostatistics at Harvard, and I've been doing clinical trial methodology research, also practiced the clinical trials for, I would say, more than 35 years. And some of my methodology developed, actually routinely used in clinical trials. And if you remember TPA's clinical trial sponsored by NIH many years ago, it's a very interesting. They use multiple outcomes, actually use a totality of evidence. So today we are going to talk about the totality of evidence from a TREASURE data. Thank you.
Thank you, LJ. Yeah, we're going to talk about that NIH clinical trial here in a little bit, but LJ, I'm going to put the first question to you. After the release of the top line data, we had a lot of people that were either writing articles or commenting directly to us or in the Twitterverse about us cherry picking results or us manipulating the data. And I was wondering if you could just spend 1 second to talk about what is post hoc analysis versus defined pre-specified analysis of a clinical set of data.
Thank you, Willie. I think the clinical trial methodology, or in general, just clinical trial, I don't think it is not an optimal way to actually assess treatment effect, including the safety. The reason I'm saying that, you picking up a primary endpoint. In this case, we call this excellent outcome, right? Excellent outcome, if you know the definition, I believe you do, it's a very, very tough hurdle to pass. Somehow you have to require the patient almost like a behave normal after those stroke. And you notice in the treasure data, 90 days, the number of excellent outcome is very small. It's no surprise because very few people can pass this really high hurdle. Well, the treasure study, picking up this primary endpoint. And everybody looking at this primary endpoint as usual, if the p-value less than 0.05, everybody jumping up and down, have a beer. Otherwise, people say you sucked. I think the clinical trial is not really only built upon a single outcome, which we call pre-specified primary endpoint. There are so many secondary, very important key endpoints we should have thinking about it. So that's why we're talking about totality evidence. In fact, I'm dealing with many, many rare disease clinical trials these days. The FDA actually emphasized how to utilize multiple outcomes instead of using a single so-called primary endpoint to evaluate treatment effect. So I think that there's a post hoc analysis. The wording sometimes is a little misleading, but unfortunately, we follow this same pathway. We sit in the box doing clinical trials. That's really unfortunately, but on the other hand, if we look at this global picture from different angles, looking at the treatment effect from a different progression domain, from a disease burden domain, you will see the treasure data provide a lot of meaningful result, how to interpret multi-cell the treatment benefit. So let me stop here, Willie.
Great, that's a great answer. Thank you very much, LJ. So in light of the fact that stroke to this day still has a very significant unmet medical burden, David Hess, could you describe to me what are the therapeutics or the interventions that have met the high bar of getting approval for treatment in the acute time frame for ischemic stroke?
Yeah, thank Willie. So there's really just really two interventions that have met the high bar, and you got TPA, which was approved back in 1996, and NINDS trial in 1995, and so that's approved by the FDA, and people argue how far out you can go, at least 3, maybe 4.5 hours, and maybe with advanced imaging longer. As most of you may know, the risk of TPA is bleeding, and the most feared risk is bleeding into the brain, which has always led to that being used less than I think most of us would like. And, still many emergency medicine doctors are still reluctant to use it. So, that kind of led to the rise, as Larry Wexler now knows, and done a lot of telemedicine, telestroke to provide neurologists, immediately to emergency rooms throughout the country and the world. And then the other treatment, which took many trials to get there, was mechanical thrombectomy, where you're basically mechanically removing the clot. And that window is variable, at least depending upon using imaging to define penumbra. May be able in some patients to be used out to 24 hours. But that's it. And so both of those are reperfusion therapies, TPA being a liquid reperfusion therapy, if you will, a mechanical thrombectomy, mechanical one. There's been many other attempts at developing neuroprotective, or in this case, I would call it maybe more of a neurorecovery or really a therapy that targets both. And despite many attempts, and many things that look good in rats and mice, it's been very hard to translate anything else to the clinic. So, that's pretty much all we have in the United States. Now, I realize in some other countries there are other therapies approved, but that's pretty much what's available in the United States.
Great. Thank you very much for kind of setting the stage there. So next one to Larry, and then after that, I'll be coming to you, Tom. So Larry, as a PI in Masters 1, and then having seen the top line data from Treasure, are there any signals that are consistent between those two trials, or any data endpoints that have you excited as a stroke trialist?
There are, and I completely agree with LJ's sentiment that we have to go beyond the top line results and look at the totality of evidence. When you look at the results from both Masters 1 and Treasure, I see things that are really very encouraging and very promising. And what I see is that particularly if you look across the various outcome measures that were used in both those trials, you see consistent trends favoring the treatment group. Not all statistically significant, but all trends in the same direction. I think that tells you something important. The second thing you see is that this difference between 90 days and 365 days. And what you see in both studies is that, for the most part, in several, if not most of the outcome measures, you don't see much change in the placebo group between 90 days and 1 year. The groups are fairly stable, and that's kind of what we expect based on the natural history of what we know happens with stroke recovery, that there's a plateau effect, and the most rapid part of recovery is within that first 90 days, and then there's kind of a plateau and very little further improvement out to 1 year. However, in the treatment group, we do see further improvements in favorable outcomes across multiple outcome measures between 90 days and 1 year that we are not seeing in the placebo group. And again, even though the differences don't reach statistical significance, I think for me as a stroke trialist and as a stroke doctor, that's telling me something. That's telling me that there's something to this treatment, that this treatment is doing something because it's widening that gap. There's further improvement that we don't see as part of the natural history in the placebo group. So those are the signals to me that there's something going on positive with MultiStem.
Great, thanks very much. So Tom, considering your expertise and the focus of your lab on recovery, trying to help stroke patients with neuroplasticity, could you discuss other stroke treatments or drugs that offer the same sort of continual improvement over an extended period of time?
Well, there really isn't anything out there. It's really interesting to note the difference between the acute interventions that Dr. Hess and Dr. Wexler have described and the progression of recovery. And so the acute therapies, TPA or mechanical thrombolysis, are functioned on the plumbing, opening the pipe. The benefit is nearly immediate. And then there is the normal process of functional recovery after stroke, which is most dramatic in the first 6 weeks, really, if you look at population studies, but it continues to about 3 months. and then it plateaus. And then at 6 months, it is essentially flat. And in fact, population studies indicate that most patients decline in the chronic phase after 6 months due to inactivity and other things. They actually lose function. And so one of the interesting things about this therapy and this trial, Treasure and then Masters, but the Treasure data was that patients continue to recover even after the phase of spontaneous recovery naturally closing. So there was still improvement that was significant on the Barthel index and noticeable in others after 3 months, when you would expect it to be plateauing and then flat. And the interesting thing here too is this wasn't a plumbing solution. We weren't just opening the clogged pipe, but instead modulating some of the things that you've probably discussed with before with the MultiStem therapy, inflammation and secondary damage. And so the time window for the therapy is longer. It's easier to administer MultiStem. And then the interesting thing about this data is it is distinct from the range of therapies out there and the fact that it promotes recovery over time, when normally spontaneous recovery itself is fairly flat and there is no therapy that has been demonstrated efficacious there.
Great. Thanks very much, Tom. Sean, welcome to the party. So next question's for you, and then Dr. Chu, you'll be on deck. So we got this question a lot, Sean. So how is it that you can give a single IV administration of cells within the first 18 to 36 hours after the onset of a stroke, yet people continue to show benefit between day 90 and day 365? So touching on what Tom just talked about, what's kind of a hypothesis for what we believe the cells may be doing?
I think this data really helps to validate what we've been understanding in the animal literature and from the animal studies that we've been doing. It really, to me, indicates that, in contrast to the acute stroke treatments of plumbing, where 90 days has been found to be a valid endpoint to look for benefit, when we think about this type of therapy, which is entirely different, we're looking at repair and plasticity. And in this respect, the brain is actively undergoing changes over a longer period of time. And so it really makes sense to me that we're seeing Treasure showing data that is replicating what was being found in the Masters trial, where people are showing potential benefit at that time point rather than at 90 days. Not that we couldn't potentially find a treatment effect at 90 days, but that we're seeing much more of that signal clearly separating out between the treated patients and the placebo-treated patients at 1 year because of the mechanisms we think that the cells are promoting. The brain is undergoing changes. It takes longer. And as Dr. Carmichael just mentioned, we don't even really have any treatments that can promote recovery in these time periods. So to me, I think this is a wonderful opportunity to be developing a completely different paradigm of treatment that we don't have right now, where the brain can be enhanced in terms of the ability to undergo repair. We just think about that from the perspective of the multitude of possibilities where the brain is not static, that it actually has that opportunity to undergo changes that would promote repair.
Great, thank you very much. Thanks for making it. I know you're in the clinic right now. So Dr. Chu, building off of Larry and Sean and Tom's comments, can you compare MultiStem's performance based off of the Treasure data and what you know about the Masters-1 data to other clinical trial outcomes? For example, the TPA trial that LJ had spoken about earlier in the NINDS trial.
You bet. The results of Treasure are tremendously exciting. And to me, they evoke strong parallels with where we stood with TPA in 1995. And that was just before the NINDS TPA trial results were announced. That year, ECAS came out from Europe, so an interesting parallel, a non-US study. And ECAS, the original ECAS trial, the ECAS 1 study, had 620 patients with acute ischemic stroke who were treated with TPA versus placebo. And that study, when it came out, was negative for the intention to treat primary analysis, but there were strong signals of benefit, and it was positive in a subset of patients that was designated the target population. So later on that year, as people have mentioned, in December of 1995, I think Dr. Hess, remembers that that's when the NINS TPA trial results broke open, and that study revolutionized the field of stroke. That's really not an exaggeration. I remember being a young stroke neurologist at the time, and my boss at the time, Jim Grata, was in a press conference along with the other NINS TPA investigators, announcing the results in front of a group of reporters, and the conference was broadcast on all the television networks. It's something that I'll never forget. Really, kind of a seminal moment in the field of stroke. And subsequently, of course, that paper, the NINS TPA trial, was selected as the most impactful paper published in the New England Journal of Medicine of the decade of the 1990s. So, I think that is a high bar to be compared to, but I'll tell you why I'm feeling very strong reverberations of where we stood in 1995 with TPA. The treatment effect size that is seen in the TREASURE trial with MultiStem therapy is comparable to the treatment effect size of TPA in the NINS trial and ECAS 3. And these two trials, the NINS trial and the ECAS 3 study, are basically the two major TPA trials in the field of stroke that effectively are the two pillars in our evidence base that really has led to, TPA being recommended in our current stroke treatment guidelines. And if you kind of look at this comparison further, obviously TPA was the first proven effective treatment for acute ischemic stroke, the first thrombolytic treatment, the first reperfusion therapy, but MultiStem is poised to be potentially the first cell therapy for stroke. As Dr. Hess mentioned, the first neuroprotective, neurorecovery therapy for stroke, first non-reperfusion therapy for stroke, and I would add the first potential treatment for stroke that could be applied beyond the first 24 hours. And, delving into this even further, if there is a difference in sort of this comparison of TPA and MultiStem, there are potential advantages with MultiStem. The lack of the risk of intracranial bleeding or other types of major hemorrhage, and the fact that potentially, more patients could benefit from treatment because we have a much longer time window of opportunity of treatment with MultiStem. So, I've used this analogy before because I think it helps to sort of encapsulate the reasons that I feel so excited. We're talking about Game 7 of the World Series here. There's really so much at stake. There's the winning run on third base with no outs. We've obviously got work still to do. And the Masters 2 trial will obviously be pivotal, a 300-patient trial. no pressure.
Yeah, great. Thank you very much. That'll help me sleep tonight. So here's a question from our friend Jeff. Hi, Jeff. Thanks for listening. He has a question. I'm going to throw this open to all of the neurologists, and then LJ, I'll come to you next. Basically says, in spite of the enthusiastic impressions of the data from the group of you and colleagues in the field, what is biotech and big pharma missing? Why is there not enthusiasm from the rest of the deep pockets in the biotech and biopharma space? What is not translating from your mouse into their ears? Does anybody have any feedback on that?
So I'll start if you want.
Sure, absolutely.
I think, we need a positive trial. I think that many people don't do what we do and look at the totality of the evidence across multiple studies, and they just look at the top line results and say, okay, it was a neutral trial. And so, to convince everybody out there, we need a positive trial. We need that finding based upon the pre-specified primary endpoint. We're always worried about bias, and that's the way you eliminate bias, by doing a randomized controlled trial, and you have a pre-specified endpoint, a primary endpoint, and you hope that that's how you get a positive trial. And I think that's what it takes to generally convince the non-experts that a treatment like this works. And of course, that's what it takes to convince the FDA. And if the FDA isn't convinced that if it doesn't get approved, then we can't get a hold of it. So that's what we need. We're certainly hopeful that Masters-2 will be that trial that turns this around. And I think once that does happen, so for example, David used the TPA example. The first TPA trial wasn't successful, but the second one was. And that turned everything around. And this is true of multiple therapies. You could look at mechanical thrombectomy. The first thrombectomy trials were negative, but then the second tranche of trials that were better designed and better targeted were clearly positive. Coronary angioplasty and stenting initially wasn't successful in trials, but then the totality of evidence was clear that this was beneficial. And these are treatments that are now standard of care. So I think once we get the right trials and we get the right results, people will look at the totality of evidence and get on board.
Any other stroke interventionalists want to add in, or has Larry cornered the market on good answers? Okay. LJ, quick question for you that we actually got two versions of this same question, and I think it's trying to help people understand how to look at the data, the top line data. Could you explain the concept of imputation in a clinical setting? Because people are trying to understand, if someone's treated and then dies at day 180, how does the data that patient or the data that patient could have presented or provided in the final outcome, how does that get calculated over the course of the trial?
Well, really, for example, the excellent outcome, or we call a global recover outcome, and which is a binary. So a patient died before reaching 365 days or even before 90 days. We essentially, that's a non-responder. So that's very easy. We don't even need to do any imputation at all. So that's a well-defined rule, right? In the clinical trial.
So basically, if I could just make sure I summarize this and say this correctly. So if we had somebody that was getting better and showing a lot of dramatic improvement, and then they unfortunately passed away for some other reason, we wouldn't obviously get them across the goal line for excellent outcome at day 90 or day 365, even though they might have reached an excellent outcome. Is that correct?
That's correct, because we actually pretty conservative in the clinical trials.
All right.
we give this guy credit. We said, sorry, you didn't make it.
All right, great. I wanted people to understand that not all aspects of trial design and trial management are in the hands of those running the trial. So with that, we got a question here. I'm going to throw this to Tom. Given that there are no material safety concerns that have surfaced during previous clinical trials with MultiStem, should we be using this as an existing stroke treatment right now compared to TPA where there are extensive safety concerns?
Well, it's certainly safer than TPA, and we have these efficacy signals, some of which are quite strong, and we have a trial with an expected lower age level, with age being a complicating difference between Treasure and Masters 1 and Masters 2. So it seems to me we need to let this have runtime, because if you start to introduce therapies that aren't proven or more definitively established anyway, you really get into a mess. And so the current testing plan is really on track to give us an answer here. There was discussion of how Treasure differed with age, and age is probably the most significant distinguisher in recovery rate and absolute level. And so we have a blip, and now we have a planned trial to really address this. And it would be probably dangerous to start instituting a therapy that's safe without further data on efficacy.
Great, thank you very much. So Sean, to you. So we know when we compare the Treasure population to the Masters 1 population that the median age is 15 years older in Treasure than what we had in the Masters trial. So we have had 2 questions that have come in resulting. Can you comment or speak about the frequency and or impact of strokes as we age?
Well, I think, first of all, age is one of the biggest risk factors of all for us. Unfortunately, as we age, the risk goes up to have a stroke. But even more to the point is that, unfortunately, when people are older, when they have a stroke, there is certainly a higher risk to develop complications. And unfortunately, in the very elderly, they have less potential for recovery and to achieve good outcomes compared to people who are at a younger age. And so what was really interesting and exciting is that, you have the substantial age difference in the Treasure trial compared to the Masters trial, and yet still we see some really significant findings. And again, also I think very exciting that people can achieve excellent outcomes at ages that we would expect there to be limited or less potential for recovery. And in addition to that, we also know very clearly that people, for example, who are over 80 years of age or over 85 have much higher risks for complications, infections in particular, or other types of medical complications. And we didn't see that increased incidence of adverse events in the patients that were treated with MultiStem compared to placebo. And it's, I think, also concordant and jives really well with the Masters trial data, in which this is a younger patient population, and there was less complications in the patients that had MultiStem compared to placebo. So the implications here is that not only could MultiStem have an effect to improve recovery overall and achieve excellent outcomes, but also that these cells could reduce the chances of us developing a medical complication after stroke. And that's possible, that can be achieved in patients who are much older. And we didn't know that, I think, and that's something that's, I think, really fascinating, that it's possible people over 80 years of age, for example, could derive those types of benefits from MultiStem.
Great, thank you very much, Sean. So David Hess, we received a question, it's pretty specific, but I think the MRS shift might be able to speak to this. Do we have proof that we save the life of any stroke patients using MultiStem. So the whole Lazarus thing here.
Well, you know, in an older population, you know, severe disability and death are rather equivalent. So I'm not sure in stroke you're really aiming to save lives. You're really aiming to save disability. And I'd have to look at the death rate. I have to go back and look at Treasure, but that's not really what we're aiming to do. We're really aiming to reduce disability and move people up to, you know, better recoveries.
Yeah, independent living, the ability to take care of yourself just by yourself without depending on others, I think is a really important endpoint. Seems like MRS less than or equal to 2 is gaining a lot more foothold as an outcome in recent years. So, okay, thank you very much. So let's see. We have a question here. Tom, I'll come to you. We'll focus on the stroke survivors here. Could you discuss, you know, at a high level, the economic and personal sort of costs associated with caring for a moderate to moderately severe stroke patient over an extended period of time?
Well, they're pretty substantial. As a matter of fact, most of the costs in stroke are chronic care costs. So I don't have the statistics straight off the top of my head, but it used to be around $55 to $75 billion, somewhere in that range. Something like 2/3 of those costs are in chronic care. So the audience will know that the major grouped outcome levels involve one measure, which is modified Rankin. And modified Rankin has specific cutoffs that attain or are directed to need for help, whether it's an assistive device or other things. And so when you start to climb that scale of higher modified Rankin numbers, you activate caregivers, assisted devices, and considerable cost. And that's primarily because these patients won't die of their stroke. They're now 6 months or years afterwards. They'll die of a comorbid or existing condition that's worsened by the disability of their stroke. So there's really a long time period in which patients are increasingly dependent. They need assisted devices. They need additional resources from friends, family, or support groups.
Yeah, great. Thank you very much. Yeah, those indirect costs, I mean, I think those are calculated when you talk about the big billions, people having to take off of work to go pick somebody up or transport somebody or et cetera. Those things add up quite a bit. Lots of people, the same question keeps coming in, and it had come in the earlier questions too. So Larry Wexler, what do you think about the use of excellent outcome as an endpoint in Treasure or in any potential trial?
Yeah, so I think that was an unusual choice, maybe not the best choice from a number of viewpoints. First of all, only a minority of people would be expected to achieve that. So there's a lot of improvement that can occur without getting to excellent outcome that you're not measuring there. And I think that, you know, that you want to see that the things besides getting to the excellent outcome measures. I think that the Rankin is, and particularly what we call the Rankin shift, is a better choice, and that's what's being used in Masters 2. It's a better choice because it covers the spectrum of outcomes, not just getting to that excellent outcome. That's great. I mean, it's great if we can get people to the point that they have no deficit and no symptoms and excellent outcome. That's great. But the other improvements, getting somebody who can't walk to be able to walk, getting someone who wasn't independent to be independent, that's important too. If you've had a stroke, those things are important as well, and change your life for the better. So looking at the spectrum of change in this shift analysis, I think is the way to go. It's the better measure of whether a treatment is effective. And I think not only is it better from a trial point of view, but it's better from a clinical point of view. It's a more clinically important and relevant endpoint. And for us as clinicians, if we have a treatment that across the spectrum of rank and outcomes causes improvement across the whole spectrum, that's really important to us as clinicians because we know that's what's important to patients. So the excellent outcome probably wasn't the best choice. I think MASTERS-2 is much better in that regard because we are using not only a better trial outcome, but a better clinically relevant outcome.
Yeah, thanks, Larry. And yeah, just one comment. I do know our friends at HEALIOS had intended to have a different primary outcome, but in discussions with the regulators, it was decided that excellent outcome would be the primary endpoint. That led someone we spoke with at Athersys recently that they felt that the cells didn't fail the patients, the design failed the cells, which I thought was a prescient comment. So anyhow, moving along, we're down to about the last 10 minutes here. So Dr. Chu, considering what's available to the stroke patients and community and the time windows, and I think you touched on this in your previous comment, what percentage of ischemic stroke patients could potentially benefit from a therapy like MultiStem in the time window we administrate?
Right. I think that's, again, one of the major potential, differences between the reperfusion therapies like TPA and a cellular therapy or neurorecovery therapy like MultiStem. About 10 to 15% of patients with ischemic stroke potentially qualify for a treatment like TPA under our current, practice guidelines. So because the time window is so stringent for a therapy like TPA, that excludes a lot of patients. And the fact that just simply by virtue of having, a 18 to 36 hour window with MultiStem, that means potentially far more patients could potentially benefit.
Great, thank you very much. So I have a question I was told I needed to forward to you or to throw out to you guys. And David, this will be you. So it asks specifically, what are the differences between Treasure and MASTERS-2 that could result in the different efficacy outcome? I think Larry spoke to it a little bit. David Chu spoke to it. Does a younger population in the US make patients more likely to have a good outcome? And are there other differences between the United States and Japan, like diet, et cetera, that could factor into the chances of success in MASTERS? So that's a mouthful, but if you could tick some of those boxes, I'd appreciate it.
Yeah.
Oh, I'm sorry. I meant that for David Hess. I'm sorry, Dave, but you could certainly chime in afterwards. He was falling asleep.
Why don't you let
there.
David Chu? He was doing so well. Let him go, and I'll follow him.
Okay,
ahead,
that's
David.
fine too. So David Chu, you go ahead, and then we'll come circle back around to David Hess.
Well, I mean, a number of the panelists have already mentioned this, and this is indeed an extremely important and potent predictor of stroke outcome, which is age. We know that the two most important prognostic factors for stroke outcome overall are age and the stroke severity, the NIH stroke scale, which makes sense. And, in the Treasure trial, you had a population of patients who were very elderly, who had moderate to severe strokes. And it comes as no surprise that it turns out that only, 10% of the placebo patients in the Treasure trial had excellent outcomes as a result. The trouble is really when you start to talk about the potential treatment effect. So not only is the overall, prognosis worse for older patients with moderate to severe strokes, but the potential to respond to any form of therapy, whether you're talking about TPA or a new cell therapy, a neuroprotective therapy, the concern is that for very elderly stroke patients with severe strokes, that nothing that you can do may really result in a major difference. And so that puts, any kind of study of a novel therapy behind the 8 ball to start off with. And the amazing thing is that in spite of these, being hamstrung by this type of study design and this patient population, that Treasure still managed to show such strong signal of benefit. And that's what's really remarkable. I mean, I might take this time to elaborate just a bit on what I said before about the treatment effect sizes that we saw in the TPA trials versus what was seen in Treasure. The mean modified Rankin scale shift or change with TPA in the NINS trial was about 0.5. The mean modified Rankin scale difference in the ECAS-3 trial was close to 0.2. If you look at just the Treasure study, the overall cohort in the Treasure trial showed a 0.2 modified Rankin scale shift for all patients in Treasure, including those 80 years of age and older. So that would mean that the effect of MultiStem was comparable to the effect of TPA in the 3 to 4.5 hour time window. And if you look at the quote unquote target population in Treasure, so those patients 80 years of age or under, and you provided some of the data in that subset of patients, about 60% of the patients in the Treasure trial, the average modified Rankin scale change afforded by MultiStem therapy was on the order of 0.4 to 0.5. Again, quite comparable to the effect size seen in the NINS trial, which is thrombolytic treatment with TPA within 3 hours of onset of symptoms.
Thank you, David. Dr. Hess, would you like to add anything?
No, I think David covered it really well. And like you said, I think the Treasure population, it's the oldest stroke population that I'm aware of. So like it's been said, to expect an excellent outcome, a bar in a population that old, I think it just shows that Japanese people are much healthier than we are. I mean, I think don't Japanese have the highest life expectancy in the world?
Yes.
So it was really really surprised to me that you could have a stroke population that old, and that made the choice of the primary outcome, unfortunate. But
yeah. So question here from Kurt. We'll address this to you, Larry. When it comes to FDA and or other regulatory approval criteria, does a drug or a therapy's lack of safety concerns lower the threshold required to bring a new treatment to the market?
I think it does. There's no doubt they take that into consideration, that something that's safe, has virtually no toxicity, has a much lower threshold for approval, as it should be. I mean, that only makes sense. However, it still has to be efficacious in addition to being safe. They will be less stringent about the proof of the efficacy side, given the fact that this treatment is very safe, but the efficacy still has to be there.
Great. Thank you very much. So Sean, if you had the choice choosing between a hard-baked excellent outcome or shifting outcomes to get more people able to live independently, which would you prefer? That's a question that we've gotten different versions of.
Well, I think, if it's a binary decision, you have to make one choice or the other. I would certainly want, for more people out there to be able to shift so that, they can achieve more. And I think that's a real benefit, for people. We just had some good discussion here. somebody going from dependence to independence, transitioning from one health state to another, I think is very valuable and very impactful. So if we could do that for more people and shift people down in health states that are more desirable, I think that would be, if given a choice, that would be the one that I would want to see more focused on, compared to getting everybody to an excellent outcome. Everybody's different. And we talk a lot about individualized, personalized medicine these days. And so I think that we should be thinking more about what is the target goal for individual people. People are different in terms of what they can achieve to get better. And I think that if people can get better to go to a lower, or I should say a better health state, but that may not necessarily be an excellent outcome as defined, by us in the scientific community. I think then I would certainly prefer the shift.
Great. Thank you very much, Sean. So we're almost at the top of the hour here. I'd like to give everybody, if anybody has any closing statements or would like to make a comment before we close things out here, I would give you the opportunity at this time. So LJ, is there anything you'd like to say from your comfortable chair in Boston?
Thank you, Willie. One thing I just like to emphasize what Sean just saying. Interesting enough, everybody knows if you have a binary outcome, right, just yes or no, or responding or not responding, from a statistical point of view, it's not really a good outcome to give us most power. On the other hand, if you have more choices, more than just binary, like we call it MRS shift. MRS shift, in fact, it's just basically you classify people to 7 categories, 0 to 6, and the lower the better. Okay, so you have the bucket, 6 bucket for treated arm, and you have placebo people also have a 6 bucket. You basically compare the frequency afterwards, after trial is done. So that's more powerful, the outcome we can use to detect the treatment effect. I'm just using the statistical point of view to say MRS shift, so-called shift model, could give us a more powerful statistical result if we still live in the p-value world. Thank you, Willie.
Yeah, thank you, LJ. Sean and Tom both sent their best. They had to log off. Larry, do you have any closing comments or anything you'd like to say?
Yeah, I just want to make one point that goes back to the very first question you asked about, what was striking about the study, and particularly to me, the difference between 90 days, the improvement between 90 days and 365 days in the treatment group. One of the reasons, there are many reasons, but one of the reasons why we've kind of taken 90 days as the standard for measuring outcome after acute stroke therapy trials is that after 90 days, so many things can happen that have nothing to do with the treatment. And so, the groups tend to come together after 90 days just for no reason related to treatment. So it's not really a fair assessment of whether the treatment really worked. So that's why we don't carry it out later. And here, even despite the fact that all of those things can happen that can mess up your experiment after 90 days, even despite that, we saw an increase in the spread between the placebo group and the treatment group, which makes it, to me, even more powerful.
Great. Thanks, Larry. Dr. Hess, last comment?
Yeah, I think it is remarkable, this separation. And don't forget that this was a relatively small stroke study of only 200 subjects, 100 in each arm. Mostly we're used to having much larger stroke trials, and you still see these signals, which are interesting, and in a relatively small trial of 200 subjects, so it's quite remarkable.
Great. Thank you very much. Last comment to you, Dr. Chu.
Last comment. Well, maybe I should try to go back to what I saw as the historical significance of what we're trying to do here. It's really you can't exaggerate how important the stakes are. Stroke research is a very, very difficult slog. But the few times that stroke researchers have hit it big with thrombolytic therapy, with reperfusion therapy, with the advances that have been made in stroke prevention, the effects on public health have been enormous. So again, I go back to the importance of what we're doing and the overarching historical significance, the large number of firsts that MultiStem would potentially represent in the field of stroke therapeutics.
Great, thank you very much. So again, thanks to everybody that participated this afternoon, either by typing in questions, listening, or supporting Athersys. Thanks to our panelists. Karen, I'll turn it back to you to do whatever needs to be done. So.
Well, thank you, everyone. I just, again, want to thank the panelists for your time and for everyone for joining. So thank you very much. Bye-bye.
Goodbye. Thank you.